All right, so we're going to do something a little bit different today. I'm going to ask for some audience participation. So if you want to run screaming out of the room, just please do it in an orderly fashion so it's not to disturb everyone else. So we're going to start with why innovation is important. So who here got into the business because they wanted to help people? Excellent. Why are the rest of you here? Okay. So innovation is important because we do want to focus on the patient, right? Every innovation means that we can create a medicine that will get to someone faster and maybe will be more effective with fewer side effects. So what types of innovations have we seen lately? So we've seen the first new lupus treatment in over 50 years to be approved by the FDA. There's actually a gene therapy out there right now on the market that cures blindness. There's other gene therapies coming down the line for other rare diseases. CAR T cell technology, we've gone from about 5-ish clinical trials 5 years ago to over 180 right now. Personalized cell therapy is on the rise, and we have a vaccine that has effectively cured cervical cancer. So we know why it's important. So why don't we do it? So in the book, The Other Side of Innovation, there are 3 traps to actually affecting change and really going forward with innovation. So what are these traps? A physical trap. You've already invested in a certain technology or equipment. So these big investments prohibit you from taking on new technology. Psychological. So this is the we can't fail trap, right? So you take what you've already been doing and you just think you can keep doing it forever. A strategic failure. This is failure to anticipate the future. So let's look at an example. Show of hands, who remembers the Kodak moment? Yeah? All right. So Kodak was insanely popular, right? Kodak was the name in photography. So where did they go wrong? Does anybody have a Kodak camera on them now? No? Nobody? Okay. So we can argue they fell into all 3 of the traps. In the physical trap, they were invested in printing and film. That was their business, and they wanted to stay with that. In the psychological trap, did you know that the very first digital camera was made in 1975? Anybody know that? Do you know who the man was working for, who made it? Does anybody take a guess? Kodak. It was Kodak. So Steve Sasson made the very first digital camera, and he brought it to his bosses at Kodak, and they said, this is really neat, but we're in the film business. That's how we make our money, and we're going to keep making our money in film. Okay? So eventually they might have realized this was a bad idea, and then you're thinking, well, they didn't look forward to the future after they realized that. They did. They also bought an online sharing site for photos. Have you heard of OPhoto? Who's heard of Instagram? Facebook? Right. So they thought of this, they didn't embrace it. So they essentially did the equivalent of sticking their toes in the pool, but we're not committed to getting wet. So let's think about us a little bit as scientists, as engineers, as thinkers that we are, which we are all thinkers, right? Some of us definitely. Okay, good. So one of the enemies of innovation in our area is budget. So how many people have said, this equipment still has years left in it, we're not getting rid of it now? Me, definitely. I've said it to my team before. Or we just spent all this money. Let me ask you this. What about thinking about the future? I have been working with a company who has this crazy process that is not optimized for what they're doing. Do you know why? It's because they developed it in old technology. And once you develop it and you get to clinical and then you get to production, how easy is it to change? You can't, right? So it's just easier for them to use this ineffective technology instead of what they could have done is develop it in something that was really made for their process. Another enemy of innovation, complacency. So who has heard, we've always done it like this and it's working just fine? Absolutely, right? If it's not broke, don't fix it. Yeah. And I've said this myself. So when you have a new employee and you're training them on how to do something and they're asking all these questions, how annoying is that? It's annoying, but what you're doing is shutting down their ability to innovate. So really the conversation should be, is there unnecessary waste in our process? Where could we be better? So you got to bring it back to the patient. You think about what we're doing. We're trying to create medicines, to create cures, to really be innovative in this space. So this one is very personal for me. So an enemy innovation is yourself and your own self-doubt. So I'll tell you, 5 years ago, I wouldn't have been up here, right? This is a big risk for me. Now I'm up here, I'm just terrified. So these questions, what if it doesn't work? Or what, I don't really know what I'm doing. These are hurdles you just gotta get over yourself. So the question should be, what if it does work? What kind of impact did that have, not only to yourself and your self-confidence, your bosses who love to, have people like that around if something goes really well, but what about the patient? Again, what if it does work? What if you can make something faster? What if you can get it out to that patient sooner? And this is something I want to touch on. So failure is essential to innovation, right? You got to take that chance. If you fail, you need to learn from it. Bill Gates said success is a lousy teacher, which is 100% correct. So people who are successful all the time, it seduces you into thinking that you can't lose. Whereas a mistake, which I make a ton of them, please don't tell my bosses, they're here, just between us, I make a ton of mistakes, but I learn from every one. So now let's talk about, I've given you all these reasons it's really hard to be innovative. That's a terrible talk. I shouldn't have started that way, but now I already did. Let's try to make it easy. So in our space, in our biotech space, bioprocessing, single-use technology is really tailored towards innovators. There's a low capital investment. You don't need to buy all of the large, huge equipment that you might need for a stainless steel reactor or autoclaves or even like large-scale BSCs, incubators, all this capital equipment. You might not need it. You can customize to a point, right? With staying within your budget. And thinking outside the bag, which, show of hands, who thought that was a really innovative title? That was me. Thank you. So the easiest way to grow innovation is surrounding yourself with innovators. So you know how we talked about those traps of innovation, people who want to shut that down. It's really hard to work with them like that, right? So you want to surround yourself with people who are innovators. And at Distech, we are very proud to be one of the most innovative companies that I've ever worked for. So they're my fourth company I've worked for. 2 of them went bankrupt. I'm hoping this one's not the same. So we'll go with that. So who is Distech? Why don't you know us yet? Well, you don't know us here in this space, but you need to, because we are extremely innovative and we're ready to go. So we've been known for innovations in dissolution for over 40 years. We own over 30 patents and counting. And we're taking all of that knowledge and reinvesting it in our future. So there's a brand new product line for bioprocessing, and we're leading by example. Maybe not the best example of me being up here all shaky and talking to you right now, but I'm doing this because I'm taking a chance and I'm ready to learn from it. We lead by example for quality, innovation, and customer service. So what do we bring to the table? This is a single-use bioreactor at the benchtop scale that is innovative because it is different. It's a bag style. So it's a bag liner type that fits into your existing infrastructure that you may already be using. So how does that really fit into, if we take it back to the traps, the innovation traps? So we want to get that cheese out of that trap without getting smacked in the back of the head, right? So the physical trap, the Bio 1 can actually use all of your existing infrastructure. So if you have a bioreactor controller, it can use that. You have a motor, we can work with that. You have a glass and a stand, perfect, we fit right in there. Utilize your existing sensors, your cables, everything you've already made that investment for, we can fit with and work with. Psychological. The Bio 1 sub can do what you're already doing, we can just do it better. So it's a better model for production scale because, look around you. Who here is building giant stainless steel bioreactors? Everyone is moving to single use. Everyone's moving to scale out and not scale up. So what you want to do is you want to do your development in a single use system, especially a bag liner system. So let's think again about that budget trap. So on the surface, you might think single use, is that going to really be a return on my investment? Absolutely. So you need to think of some of the hidden costs of production as well. Your utilities, the downtime, that's the biggest. If you can squeeze an extra 2 or 3 runs out and get more experiments for your product, why wouldn't you want to do that? Everyone wants everything faster. If you're using subs at the production scale, you want to use a bag liner style. You don't want to use glass or polycarbonate. And we have the data So we have the data here showing our comparability with the glass. We have definitely more in-depth. I don't want to take you through all of it right now. But definitely come visit us at our booth. Visit me if you enjoy this at booth 1079. So let's go back. So how do you be innovative and how do you make it productive? Find a pinch point, look for a solution. So this is your biggest takeaway from this is don't just try to innovate to be innovative or say you're innovative. Find a solution. So complacency will cost you. Doing things the same way all the time, and it'll cost you in ways that you haven't even thought of before. Your decreased production. So all of your competitors, they're trying to be innovative and they're trying to get more out. Employee satisfaction. This seems like a strange one to bring up when you're talking about innovation. It's not. So how expensive is it to train up a brand new employee? Hugely expensive. How expensive is it to retain talent? Pay attention. Give them some space to be innovative. You don't want to play catch up with safety or quality standards. So we'll go into a little bit about some of the safety considerations when thinking about single use, but definitely with your quality and your safety, you want to be top of the line. And you want to be able to advertise that to your customers or to your patients or to your quality team. And you don't want to play catch up with the leaders in your field. You don't want to be the last one to transition to a single use solution. So I like to call this my how to make friends aspect. So I came from manufacturing. So is anyone else in manufacturing ever? All right. Have you ever cleaned a BSC while you're fully gowned up? Oh, that is sweaty, right? And you know what they do back there, what I've done, is you get real hot, you just take IPA baths, right? If you haven't done it, try it. It's great. If your air conditioning is broke at home, take a bottle of IPA with you. Don't steal from your company, okay? So close the seed trade. When you're in manufacturing, cleaning a BSC, the amount of cost it is for the time, the changeover, the cleaning materials, you actually charge you for those little sterile wipes. It's unbelievable. Close the seed train. Eliminate all that cost. Also, you will limit open processing steps. Every risk assessment you're going to do in manufacturing, where are your steps open? Where's your product open to the environment? Even if it's a Class 100 environment, where is it open? You're looking at long-term cost savings. A fully closed system, you also have the possibility of eliminating changeover. Eliminating changeover saves time, saves paperwork. You make even more friends. I have so many friends I can't even count. Just kidding. Part 2, high density cell bank. So why are those in quotes? Because some people won't want to call it a cell bank, they'll call it a process intermediate to avoid all the regulatory implications with a cell bank. Perfectly fine, you can do that. You can keep your cell banks little, make a high density bag style cell bank. Skip the seed lab altogether. Freeze in the bag, thaw in the bag, inoculate directly into a sub. Yeah, we like that one, right? It's a good idea. I wish I could claim it. I think I found it on the internet. So alternatively, you can make smaller high density cell banks. If you don't have enough cryo storage for a large storage for a cell bank, make a smaller one. Grow it in a 5-liter sub at a small volume, feed it up, and then take that and the high cell density, inoculate it into a larger sub. With all of the high turn-down ratios we have now, we're getting better and better. So you should really be able to do this. Mitigate risk for production. So if you were doing all your development in a glass and stainless steel or even a polycarbonate system, how do you know what it's going to do when you get into a bag liner? We were just talking about how every CMO, all the processing around here is moving to single-use, large-scale production. The worst thing that could ever happen is you optimize this process. It's your baby. You've been working on it for years. It's in glass. It's in stainless steel. You take it to a CMO, and it crashes. And you don't know why. Cells do react a little bit differently to bag liners than they do to other materials. It's a known fact. So you think about the costs that you just put in to doing all that work, getting a CMO or scheduling time in your pilot plant. Also, does anyone here like to write deviations or investigations? I was really hoping to find someone so they could write all mine for me. I'll continue to look. The cost of an investigation is astronomical when you think about personnel time. So you want to know exactly what your normal operating ranges are going to be in a backstyle liner before you get to production, before you get to a late phase clinical trial. Safety, safety, safety. This is so important. I say it 3 times. You want to keep your people in the exact same condition when they leave work as what they came in. Maybe a little bit more tired and sweaty if they're cleaning all those BSCs, but you want them to remain safe.